I'm going to talk pretty much just about one project, but it was a very, very large project. It took a very long time. So I'm just going to focus on that. It was this project with Stuart and his posse, physical ecology lab in the UK, on just curved bacteria and why they might exist. Why are a lot of bacteria curved and not some other shape? And I did a bunch of numerical modeling of this and even got some optimality theory in there as well. So the whole we were paid through this big grant, and the grant was just about why bacteria have these really diverse shapes. So if you read through the medical literature, you might be fooled into thinking that bacteria are either spheres or rods or maybe once in a while some kind of spirochete shape. But actually, if you look hard enough, there's just every shape you can imagine: squares, stars, weird sort of branched shapes. And there's this great paper by Young, and this great quote, which is about most people not really caring very much, it would seem, about these shapes, even though bacteria seem to have evolved these shapes very purposely. So what I started to think about at some point were these comma-shaped species, these vibrios and other things that are curved rods. And it turns out in the ocean, I have a lot of experience in oceanography departments and such. In the ocean, most motile bacteria are either straight or slightly curved rods. So why are there so many of these things out there? That's what I wanted to ask. So, this is just a smattering of different species. A lot of these are marine. Maybe all, I don't know about all of them, but most of them. And of course, some of them are only slightly curved, some of them are more curved, some of them are almost semicircular even. But why? And because this is, of course, is a fluid mechanics seminar, maybe it has to do something with how they swim, their motility. Maybe it helps with their motility. So many bacteria that swim, they use rotating propellers, flagella, that are more or less rigid. So on the left, there's a good example of this helical-shaped propeller-like flagellum. Some bacteria have one of these, sort of a simple case. Other bacteria, like E. coli, have a whole bunch, but luckily, they tend to form a bundle that looks like kind of one fat flagellum. So in my modeling, I've always just assumed there's one of these propellers behind the cell, and you could say, well, if it was something with a bunch of flagella, as long as they were in a bundle, we're more or less okay in that approximation. But again, the question is, like, how does the morphology matter here? Does a curved cell body, have some effect versus a sphere or a straight rod or something else? So the thing is, form and function in these microscopic swimming organisms is really barely studied. So there is a large body of work focused on the tail shape, the flagellum, but there's a lot less work on what the body of the cell looks like. So cells are usually modeled, well, often they're modeled as points even, maybe a spherical body in a lot of work. And sometimes if you want to get really fancy, you use an ellipsoidal body of some aspect ratio. So here's a paper by Shum Gaffney and some guy, DJ Smith, from a while ago, where they looked at a bunch of different things. But one thing is they took some species of real bacteria and they approximated them using these ellipsoidal bodies. I know that things like swimming efficiency, comparing across different species, but if you look at Vibrio cholera, it actually is a curved rod. It's not an ellipsoid. So again, the question is, like, what if you actually start to think about these more complicated or realistic shapes? What effect might that have? And my hypothesis was maybe that curvature can actually give them some kind of enhanced motility, some kind of enhanced speed, basically, swimming speed. I'll get to why I had that hunch, I think, in the next slide. So, so there's an very old paper by Fan Tien et al., where they looked at ellipsoidal body swimmers, and they just tried to find the optimal shape of both the body and the tail behind it for swimming efficiency or swimming speed for a given power. And they found that the optimal shape was this. It's hard to see here, but if you imagine this is a sort of a general triaxial ellipsoid that's squashed sort of from left to right. So it's actually not rotationally symmetric. It's squashed left to right here, and that's the best swimming ellipsoidal cell body. And why is that? Well, the video that I had most easily at hand was one I made myself when I was just playing around. I'll get to the modeling in a second, but this is just a simulation of this weird coin-shaped bacterium. And so the idea here is, because you have to have a balance of torque, the body you can see is rotating one way, and the tail has to counter-rotate the opposite way because this thing has no external forces or torques on it. So because of that constraint, ideally what you want here is for the body of the cell to have very high rotational drag but low translational drag forward. You want it to go forward easily, but you want it to be really hard to rotate that body around the swimming axis, because if the body rotates really fast, that means the tail is going to rotate slower, and if the tail rotates slower, it's not really going to go anywhere because the tail is actually what's pushing this thing ahead. You want the tail to rotate as fast as you can make it rotate relative to the body. It's this weird sort of constraint on swimmers with rotating tails, like bacteria. So I had this hunch that, well, maybe if we imagine a curved rod on the bottom right here, and you imagine it swimming more or less along its tail axis there, its flagellum axis, well, as it gets more and more curved, it's gonna be harder to go forward. It's gonna increase translational drag, but also it's gonna increase the rotational resistance as well as it curves out from the swimming axis. So depending on which of those increases more, efficiency might improve, it might not improve. We don't really know. We have to crunch the numbers and see the balance between translational and rotational drag. So that was my hunch on why curvature might help. So I numerically modeled Stokes flow around a curved bug. So in the Stokes flow regime, there's no pesky nonlinear inertia terms in Navier-Stokes. So you just have the Stokes flow equation and conservation of mass that you have to solve. And in the course of modeling the fluid flow around a swimming curved rod, it turns out we're going to also solve for how the swimmer itself is going to move through the fluid and things like how fast it's going to move. So I used the boundary element method, but I used a regularized Stokelet version of it. So in general, the boundary element method, its big advantage for these low Reynolds number flows, these creeping flows at small scale, is you don't have to discretize a whole volume of space, like let's say finite elements or something. You only have to discretize the boundaries, the surfaces. So in our case, because I don't have any walls, I just am modeling these swimmers in, say, the open ocean. We only have to discretize the surface of the cell body and also the tail that I'll get to in a minute. So we have this boundary integral equation that relates the velocity at any point in the flow. Here we're gonna write down velocities on the surfaces, on the nodes of the mesh on our swimmer, and they're related to the integral of this Stokelet or regularized Stokelet kernel dotted with this F. F is the traction, which is just the surface stress of the water acting on the swimmer surface. So you have to integrate this S dot F over all the boundaries in the problem. So over our entire swimmer in our case. So in the regularized, all right, the issue with boundary element methods is this singular Stokelet kernel is kind of a real pain. So you have this singularity you have to try to integrate over. It's a real pain to work with. So luckily, Ricardo Cortez came up with a regularized Stokeslet um, version quite some time ago. And so you can smooth out this annoying singular behavior, and you get this sort of blob-shaped function instead. So a regularized version of S, and it has some regularization parameter epsilon. And as epsilon goes to 0, you recover the original boundary element method. But if you have a finite epsilon, it just makes your integrals much easier to do numerically. So in my case, my epsilon was very, very small actually compared to the length scale of my body, of the bacterium, compared to the mesh, and so on. But it still made the integrals much easier to do numerically. So anyway, the meshing, for some crazy reason, I decided I just had to have these curved triangular meshes. So there's second order triangles here, six node triangles. Sorry I didn't show the little dotted nodes, but there's 6 of them on each triangle. So not only did I fully mesh the bodies of all my bacteria, but for some crazy reason, I decided to mesh the tails too. So everything's covered in these curved triangular meshes. And so because I had these very refined, fancy meshes, you might say, oh, the code must have been super slow. Sort of. It wasn't the fastest, but there were some improvements to be made. I was able to parallelize things over all my, so you write one of these boundary integral equations for every node on your mesh, and there's a lot of nodes. So it makes sense to do that in parallel if you have a bunch of CPU cores floating around. Also, a tweak I made was to do these spatial integrals, I used adaptive integration. So if over one of these triangles, there's a lot of crazy variation going on in this integral, you can keep subdividing it into smaller triangles and get as accurate of an integral as you need. Finally, in case anyone out there is a big fan of MATLAB, I'm a huge fan of MATLAB. People say, oh, MATLAB's slow, that's the real problem. But there's actually a toolbox called Coder that's pretty awesome. You don't even have to know C or C++. I barely know it, but you click some buttons and you fix certain parts of your code that it complains about, and all of a sudden you get a compiled version of your code in C that's about 20 times faster, at least for me. So that was a huge speed up. So I highly recommend looking into that if you're a big MATLAB user. So how are we going to solve for how this bacterium is swimming through the water? Well, we prescribe just the rotation of this tail. I'm going to say it's rigid, which is a decent approximation for bacterial flagella. And the way we solve for how it's going to swim is because we know there's no external forces or torques on this thing, we can say the sum of the forces is 0, the sum of the torques is 0, and we're going to set the torque between the two. We're going to set what I call the motor torque that's causing this tail to rotate. And what we can do is we have our boundary integral equation, and for U, we can sub in what we know to be the case because the body and the tail are both rigid, right? So they're governed by rigid body motion. So actually, you only have a U for translation and omega for rotation that governs how both of these things are going to move. You don't have to have a separate U at every node on the mesh. So what we're going to solve for is this traction, the surface stress all over the swimmer, and we're going to solve for, over time, the swimmer position, let's say the centroid of the body or something like that, and this orientation, 3, let's say, Euler angles for how this thing is situated. And Valerie is here. Valerie is here. And there's some details there because so our rigid body kinematics were in a moving frame attached to the swimmer, but our final answer has to be in the fixed frame. Our x and theta are in the fixed frame. So there's some mildly unpleasant details, some unpleasant rotation matrices there that I won't bore you with, but they're in the paper. So this is probably the last detail of the method. To speed up the time stepping, there's a nice trick here, because if you were this bacterium swimming through, as far as you are concerned, if there's no walls or anything, everything always looks the same to you, right? Like it's always going to be, there's going to be a lot of symmetry after your tail goes through a rotation. So because you have to be quiet, I'm doing the show. So because everything is periodic in this moving frame, we only really need to interpolate these U and omega in the moving frame over a tail rotation, just one rotation of the tail. So you only really need, let's say 4 or maybe 8 max points to get really accurate. So this blue curve, the blue dashed, I only have 2, a measly 2 points along a tail rotation. But as soon as I put 4 in, I get to that solid curve, and the difference between 4 and 8 points, you can't even see it. So using these sort of trigonometric interpolation functions is super efficient for this sort of problem. Now, if you put a wall in, this all breaks, but no walls here. Okay, so here's another gratuitous video of just a spherical swimmer moving along. So what we can do is define a swimming efficiency basically the same way most people do or have with the power. Okay, you have to be quiet though. Sorry, everyone. So you can define the power that the swimmer is putting in is just torque, that motor torque that we set, times how fast the tail is rotating. And if you know how fast it's swimming, if you square that and you have a sort of a size parameter in there, you get a non-dimensional swimming efficiency out of this. So to validate the code, I compared to that paper by Schuech et al. I picked their what they call their power optimum. So this bottom one was the most efficient swimmer they found within their parameter space. And so if I model that same shape, that same body and tail, I got their same power efficiency to within about 1%. And this was just a completely independent code. So I trusted that as validation. Here are some more gratuitous videos of actual curved rods that I started looking at. So we got sort of a long, slender, banana-shaped cell. Then we have these higher levels of curvature. Sorry, this is very glitchy for me. I don't know why. That's my computer. But there'll be more videos like this. So I basically wanted to start to do, a lot of runs like this and try to figure out, can I find an optimal curved rod body shape that's the most efficient swimmer? So I have to get serious now and start to look over a whole parameter space of different shapes, right, to actually figure out if there's the most efficient one in there. So I have this whole grid now I've created of curved rods. So along the x-axis, I have elongation, just how long and slender the body is. And up the y-axis is what I call K, very loosely curvature, which is how much of a sort of donut the cell body makes. And I should mention that all these different cell bodies are the same volume. That's what's constant. So if you want to get longer, you have to get skinnier to preserve volume. So all of the work now I'm going to show is over this morpho space of curved rods, going along the x-axis just being straight and increasingly slender, and up the y being more and more like a donut. And of course, there's some missing space in the upper left because you just sort of start to self-intersect over there. So it turns out I just showed all the body shapes, right? But what about the tail? Obviously, the tail is what's making this thing swim. So this was the pain of this project was the tail. Because if we want to compare different body shapes, the thing is, each body shape is going to have some tail shape that's the best tail shape that goes with it. And the best tail shape for a spherical body is not necessarily going to be similar to the best tail shape for, let's say, a curved rod. So I had to go through all those different body shapes and find the best tail that goes with it in order to avoid any bias in the results. So the tail has three parameters: amplitude, wavelength, and how long the whole thing is, number of wavelengths. And so I had to optimize these three parameters for everybody. Huge amount of work, but that's what was needed to avoid bias. The interesting thing about this is here are the sort of landscapes over elongation and curvature of the body for the best tail shape. So you see the amplitude and wavelength, the numbers are different, the values are different, but they look qualitatively the same exact pattern there. And if you look at number of wavelengths, how long the tail is in total, there's not too much variation in that over all the different body shapes. So over here, I've shown you the best tail shape for a sphere and the best tail shape for this sort of semicircular, long, skinny curved rod. And actually, it looks like you just scaled up the one on the sphere, doesn't it? They look basically the same shape. So this is sort of a future question that is worth looking at. Is maybe this optimal tail shape actually is constant over all different body shapes and be any body shape. I don't know. Maybe the tail just gets bigger as your body kind of gets bigger in some sense. Anyway, we found the optimal tails for each body. So now we can actually rescale our results. So we didn't know how much power each cell was going to use in the simulation, but once the simulation is done, we can rescale, assuming that all the cells use the same power, and then we can figure out how fast they would swim if they all use the same power, and we can race them. So here is the somewhat, I don't know, famous / infamous swimming race between just this random group of shapes. So they're actually ordered. The fastest one is at the top. The slowest is at the bottom. Most of them are really almost the same speed, honestly. The only one that's really much worse is this lower, long, skinny straight rod. And it's so slow because when it's long and skinny, it's very easy to rotate that body, isn't it? It has very low rotational resistance, and so the body down there is rotating fast, and the tail is not rotating as fast as it could. That's why it's so slow. But not a huge amount of variation, right, in swimming speed, swimming efficiency here. But still, over evolutionary time, maybe small differences could still be important. So what I then did is, so I'm going to show a lot of these landscapes, what I call performance landscapes. So here I've just plotted the swimming efficiency for all these different body shapes. Again, each body shape is paired with its best tail, which I'm not showing here because it'd be super cluttered. But there's a lot to unpack in this plot. So again, we see the lower right. The only really bad shapes you can be are these straight, slender rods. But everything else is like within maybe a few percent swimming efficiency. They're all really close to each other. Now it turns out the best possible shape here is this dark pill shape down here. That's the best one over this whole morphospace. So kind of boring. The best one wasn't curved, it turns out. But there's this cool local max up here, which is a semicircular curved rod. And this one is only, let's see, it's only 0.3% different, this local optima versus the global down here. So there's very little variation over a huge swath of parameter space. And what is interesting is if a cell does want to elongate for some other reason that I'll get to very soon, then let's say you want to be an aspect ratio of 8. Well, if you're straight, you have a pretty big penalty to efficiency. But if you get curved, if you move up, you really mitigate that quite a bit, and you have almost no penalty anymore, but you're still elongated. So curvature can help. It just can't be the only thing explaining what's going on. Now, anyway, what is going on? Where are real bacteria here? I didn't talk about the actual shapes yet. So you have to take a detour. It turned out to be a rather long and somewhat painful detour because we needed to go through the literature and figure out what shapes are out there. There was no database of quantified curved rod morphology. Sure, there's the paper will say it was slightly curved, it was comma shaped, it was very curved, but that doesn't mean anything, of course, mathematically. You need to put a number on it. So we, and by we I mean Taddy, which we couldn't have done this without, she went through and painstakingly with this microbe J plugin from J scrounged images, mostly from Berge's manual, these huge tomes of all different bacteria, and also just straight up Google searching curved bacterium. And you find papers that they found a new species, and if you're lucky, there's one image that you have of the species, and that's what we had to use. That's the best we can do. We did the best we could. So we quantified the elongation and curvature of about 5,000 cells over 360 images. We had about 200 species when all is said and done. And over here, I've plotted this cloud of dots that we get over our elongation curvature morphospace. There were these two wacky outliers out here. They were really long and skinny. Methanospirillum is an aspect ratio of over 20, this sort of needle-like curved cell. But it'd be very expensive to simulate that cell shape, so we had to cut things off somewhere. We cut them off at an elongation of 10 going forward. So here's that same morphospace again, just cut off at 10 and made to look a bit fancier. So I put in a few actual species on here. So you get a lot of density of species in this sort of what you would expect as a curved rod, a slightly curved rod, not too elongated, run-of-the-mill Vibrio, let's say. Over here, it's most of them. Some of them are more elongated, and a few are more curved, but there's really no like donut shapes out there that swim. The most donut-like is the semicircular Leptospirillum, which I'll talk about at the very end as kind of an outlier. But you don't really get very high curvature, at least in motile bacteria. So anyway, if we were to superimpose these real shapes on our swimming efficiency plot, if you squint and compare, there's not really you wouldn't say that all of our real species are, let's say, within one of these contours or clustered around one of the optima or something like that. They're just kind of all over the place. So we haven't really explained why these different shapes exist with swimming efficiency. There's no clear correlation here. So the thing is, why do bacteria swim? Why does anything swim? They're not swimming for fun. They're swimming probably to find food. So here's a video by Smirga et al. From the Stoker group. And they sort of exploded this diatom with some kind of intense pulse of light. And over time, you see bacteria buzz in and form a dense kind of buzzing cloud of, looks like you'd think this might be flies. It's actually bacteria that are eating up whatever has the lifeblood of this diatom, right? And so what they're doing is chemotaxis. They're sniffing out whatever is coming out of this diatom and racing toward it as best they can to eat. Whoops. So how can we model this chemotaxis that might have shape effects also? So if you imagine a bug swimming through a chemical gradient, it's taking samples. We're going to assume it's taking samples over time, temporal sampling. So take sample 1 and sample 2, separate over delta t, and we can compute from a bunch of work that Dusenberry has done some time ago, a signal-to-noise ratio for these measurements of the chemical gradient that the bug is trying to go up and go toward the source of this food. Or at least we can talk about the signal-to-noise ratio that depends on the things that the swimmer can control. Of course, it's going to depend on environmental parameters like the steepness of the gradient, but we're just going to look at what the swimmer has control over, what shape might affect. So we know that the signal-to-noise ratio for chemotaxis is going to depend on the distance, LS, between the two samples, and it's going to depend on this tau. Tau is how long it takes before the swimming direction of a bug is just randomized due to Brownian motion. So this LS is easy. That's just going to depend on how fast the bug is swimming. So the faster you swim, the more distance you're going to have between your samples in a given delta t. The tau, so it has to do with Brownian motion. Here's an example of some dead or at least incapacitated E. coli just bouncing around due to Brownian motion. And if you squint, you will hopefully notice that this elongated cell is bouncing around a lot less. It's sort of staying in its same orientation for a lot longer than this short cell over here that's very rapidly spinning around. So if you're a cell trying to track food or something like that, you don't like this because when you can't figure out which way you are headed, there's no point in really in doing anything anymore, because how are you going to track a gradient if you don't know which way you're going? So chemotaxis is going to depend on both how fast you can swim, swimming efficiency, which we already quantified, and also this Brownian rotation, this tau, which is going to depend on shape. The longer and skinnier you are, the more resistant you are to Brownian rotation, and the longer you can swim straight for. So more pain. So because we're thinking about now a new kind of task, we're thinking about this product of swimming speed and tau, the time scale for Brownian rotation. Technically, the optimal tails are going to change now from what they were for swimming speed or swimming efficiency. So you have to optimize them again for chemotaxis. So I did that very painfully with many CPU hours. Now it turns out that as you make your tail longer and longer, you just get better at this chemotaxis because you just can swim straighter and straighter, or straighter for longer. Even if you swim really slowly, doesn't matter, because you swim straighter for longer, that wins. So what we had to do is cut off the max tail length at some reasonable value that you see for real bacteria. And then we optimized within that. So here is optimized within some max arc length for these two different shapes. And you can see there, things are still changing. So tail shape is still important here. So again, we optimized again for every possible body shape to be fair, to reduce bias. And here's our landscape for chemotaxis over all our curved shapes. So what's going on? The worst you can be is basically a sphere, because you're just gonna lose your orientation really quickly. You're not resistant to Brownian rotation at all. And as you get longer and skinnier, you swim straight for longer and longer, and chemotactic ability improves. But what's interesting is, yes, long straight rods are most resistant to Brownian rotation, but you get that boost in swimming efficiency if you're curved, remember. The worst swimmers are down here and straight. So because that curvature increases your swimming speed, your optimal shape is going to be very long and skinny, infinitely long and skinny, but curved also. Still, if we put our real shapes superimposed, imagine superimposing them on this landscape, there's not really a clear pattern here. It's not like all our shapes are super long and skinny and curved. No, they're actually opposite. They're all over here in the blue. So we haven't really explained why bacteria are curved with chemotaxis either, at least not by itself. That's clear. So keep skipping. So what else? What other tasks might these bacteria be optimal for? Well, okay, you're chemotaxing toward a food source. That's not enough. You actually have to be able to eat the food. So if you think about small molecules that have to diffuse into a cell in order to be quote unquote eaten, we can call that nutrient uptake. And so I modeled that actually here in COMSOL. It was just the lowest, the easiest thing to do. So I just stuck in a very large box, or I think actually a huge sphere, a spherical domain, one cell. I didn't include the flagellum for simplicity. I just set a boundary concentration of 0 on the cell surface and a concentration of 1 infinite or far away on the surface of the domain. And of course, things are going to get sucked into the cell then. They're going to diffuse in. So I just looked at how much, how well these different shapes can uptake nutrients by diffusion. So you might expect that getting really long and skinny is going to increase your surface area to volume, and it's going to maximize your diffusive uptake. And indeed, the best shape here is infinitely long and straight and skinny. And that's the most important variation, is elongation. If you start to curve, you get a little bit of shielding in the middle, don't you? If you have a donut shape, that inner part is a little bit shielded versus the outer part. So curvature doesn't really help you here. Curvature is not a good thing. It's not hugely important, but it's not good. So anyway, can real bacteria be optimized for nutrient uptake? No, if this is not at all really useful, is it? Because they never be curved. They would be straight if they want to maximize uptake. All right, so another task down. Let's try another one. All right, so cells eventually have to turn. If they decide they're going in the wrong direction, they have to go a different way. So here's some E. coli tumbling. They tumble by rotating their flagella in the opposite sense they usually do. The flagella kind of splay out all over the place, and they sort of randomly turn the cell around. And even cells with a single flagellum can do a different thing. They can flick and reorient like that. So I figure, well, I don't know, maybe bacteria actually want to reorient really easily. Maybe that's what they care about. So I call this tumbling ability. And if the flagella are kind of randomly buffeting the cell, I assume that's kind of like Brownian motion. So again, I looked at this time scale for loss of orientation, but here I actually just inverted it and I said, well, to tumble easily, you want to be a sphere. You want to be really easy to reorient if you want to tumble easily, right? If you're really long and skinny and straight, it's going to be very hard for you to tumble or reorient on purpose, and you actually will never be able to go in a different direction than you were really going. So that's not great either. So okay, maybe bacteria want to tumble. Well, if you remember what that cloud of data points looked like, I mean, they weren't all spherical, were they? They were moderate elongation, moderate curvature. So maybe this is one of the pressures on cells that they're going to have to be able to tumble to some extent, but certainly real bacteria aren't optimized just for tumbling. So you can see this pattern. We sort of keep getting a little bit of the answer, but certainly not the whole answer to why these curved bacteria exist. So the final task I thought about is not even fluid mechanics related. It's just how difficult is it probably to build these cells from scratch. So presumably there's some cost to having crazy wacky shapes with sharp kind of curves or sharp corners and things like that. So I assume there is some cost to both elongating and becoming more curved. I assume that the simplest shape you should be able to have is a sphere, basically, right? Because that's what you'd get if you just have like a lipid bilayer, you'd get a sphere. So we looked at all different cost functions, actually, but due to time, I'm just going to talk about the most kind of, well, the one that worked and the one that's kind of the simplest in a sense. So if you look over a surface of your cell body, you could define these principal curvatures mathematically. That's just sort of how sharp the surface is at a point. And there's something called the Gaussian curvature, which is the product of these two principal curvatures. But because I assumed that a kind of convex curve would be equally costly to a concave curve, right? Like whether you have a sharp corner or a sharp kind of inward, sharp pointing out corner or sharp pointing in corner, they're probably both going to cost. So I took the absolute value of the Gaussian curvature to penalize anything that wasn't smooth, basically, no matter whether you were poking out or in. I integrated that thing over the body surface to get a single number for how kind of sharp the body is in general. So this integral is going to penalize the quantity elongation times curvature. And the only issue with that is that if you have a curvature of 0 along your x-axis here, along your elongation axis, you actually would just have a 0 for this integral, and we don't really want that because we want elongation to actually cost you something also. So the way to get that in there is you can multiply by the area, the surface area of the body. And so, of course, as you get longer and skinnier, your area increases, and so you're going to have an overall cost associated. We have a power of a half there here to just Dave maybe put that in to fix some intuitive scaling, but it doesn't really affect the results in the end. So anyway, of course, bacteria aren't all spherical, I know, but maybe it's one of the pressures on them, right? It's logical to assume it's going to cost them something to build one of these donut-shaped bodies. So anyway, go back to our full morphospace now. I put in the optimal shapes for our various tasks. So swimming efficiency had a local max up here. It had the global max down here, a capsule shape. For construction cost and tumbling, the best thing you do is a sphere. And then all the way at the highly elongated zone, the best thing you could do for nutrient uptake was a straight skinny rod. And the best you could do for chemotaxis was a slightly curved, very skinny rod. And so here are all our real species. And what's interesting, and what I started to wonder about, is, well, they're all kind of in between these kind of vertices almost. It's almost like a triangle here, and they're all in between. So maybe all these species are somehow optimized for a bunch of these things at once. So how can we explain this whole diversity? So very luckily, I just happened across a series of papers in the Alon research group. So this paper by Cheval et al. In Science was about just applying Pareto optimality theory, which is really from economics, to biology and to evolution. So what they did here was they looked at all these different organisms, finches, the beak shape of finches, the body shape of ants, and the wings and the body shape of bats. And they defined their morphospaces with things that were obvious and easy to measure. So okay, beak shape and size, the head shape of an ant, and some aspect of its body shape here. And you get the data points, the actual shapes of all different species that can tend to be within these sort of triangular regions of the full morphospace. And what they did is they plotted their actual phenotypes, and they drew a triangle around it, and they looked at the corners of these triangles, and they said, well, we know a lot of things about these species. We know that this bat over here, we know what it eats, we know sort of where it tends to fly, we know a lot about the ecology of each species, and they just took good guesses. They inferred about what these corner, what we call archetype shapes are optimized for. So that's how they concluded the trade-offs that were probably influencing or constraining the evolution of these different types of organisms. Now the problem is that for bacteria, we don't know a whole lot about the ecology. Like you can move from one sand grain to the sand grain next to it and have a completely different micro niche, maybe a slightly different oxygen concentration or something, a whole different set of bacterial species on it. So it's very hard for us to look up data on different bacteria and see what these tasks are probably they're probably optimal for. So we had to go about this a different way. Also, our tasks, well, you'll see we tend to not get triangles actually for our bacteria. We have a more complicated situation. So what I did is sort of a brute force strategy here at getting that Pareto optimality trade-offs. So the overall problem here is that your overall sort of ecological fitness is going to depend on all different things. It's not just going to be swimming efficiency or just chemotaxis, just construction. It's going to be some combination, of course. We don't know the weights of each of these tasks. So this Pareto optimality theory, the solution here is you just have to assume that the rank matters. So we don't know how important swimming efficiency is in the grand scheme, but we know that if you go from a swimming efficiency of 1 to 1.01, that's got to help in some way. We don't know how much it helps, but it's got to help. So we can say that a shape, we can compare one shape to another here, or one shape to all the other shapes, and we could just say a shape is suboptimal if you look at all the tasks that you think are important. If that shape is beaten by something else in the morphospace, it's beaten by all the tasks, it can't be very good. The other shape's got to be better. So if a certain body shape cannot be beaten at every task at once, then it's Pareto optimal. That's all it is. So if we imagine this donut-like shape up here at the top, and we imagine comparing it to something that's less curved, lower. So imagine going down here on the swimming efficiency graph. You're actually going to improve swimming efficiency here going down if you squint. If you go down in curvature for chemotaxis, you're going to improve a little bit. And if you get less curved as far as construction cost or ease of construction, that's also going to improve. So why would you be this shape if you can be down here and be better at all the tasks at once? Of course, you're going to do that. That's why this donut shape is suboptimal, and this shape down here happens to be Pareto optimal, because if you move in any direction away from this shape, you might improve in one task, but you're going to have a cost in a different task. So we kind of just brute forced compared all these shapes to each other and figure out which are Pareto optimal and which are not Pareto optimals, which are suboptimal. So we didn't really know which tasks are important. So we just brute forced it. So we took all different subsets of the tasks I went over: the swimming efficiency, chemotaxis, tumbling ease, construction cost. So we had to take at least 3 at once, because if you just have 2 of these tasks trading off, you can only get a one-dimensional curve of optimal shapes between them. And we know our complete set of data points spans this large 2-dimensional region. So we took at least 3 tasks at once, and we went up to all of the tasks combined. I think up to 5 or 6 tasks. We said, well, maybe they're all important. And when we computed the Pareto optimal regions, computed by brute force, and we compared that to the actual kind of convex hull of all our actual shapes that we see in nature, and we just figured that the task trade-offs that were the most evolutionary sensible, important, would match the data the best. So we had this goodness of fit parameter that just how well they overlap. The optimal region theoretically and the actual region bounded by the data. And so we compared all these. So the upper left happens to be the best. So these are the 5 best fits of all something like 38 possible task sets that we looked at. The best one is this upper left. Goodness of fit is 0.84. The next best was over here, 0.74. Visibly, it looks like this is indeed a better fit, right? There's more overlap. So I just cleaned that up a little bit here, smoothed it out, colored it in for giggles. And so this trade-off here is between swimming efficiency, the construction cost that favors a sphere, and chemotaxis. So this is the actual Pareto optimal region you get, smoothed slightly. And these are all the data points that we measured. And we have this little island up here, which is kind of interesting. This optimal island. You don't necessarily have to have a single connected optimal region if you have complicated task landscapes. But anyway, I colored in based on sort of the various weights that each species might be given to each task. So if you're all the way over here, you're very elongated, you're going to have a very high weight given to chemotaxis. So presumably, if you're long and slender and a little bit curved, you are heavily weighting chemotaxis in your sort of evolutionary history. Whereas if you're closer to a sphere, you maybe just want to be really cheap to build. You don't care that much about chemotaxis. And if you're way up here, maybe you care a whole lot about swimming efficiency. Well, maybe you care a lot about swimming efficiency as well as chemotaxis. So it's a 3-way trade-off that you get. And of course, this fits the data really well, it turned out. This outlier dot up here is that leptospirillum ferrooxidans, it's called. And what's interesting is it tends to actually wrap itself around, I don't know the exact biology, but it seems to wrap itself around, iron particles to live off them, but to get energy from them. So that could be, obviously we didn't consider that as a task. So that could be why this one is a bit of an outlier. This evolutionary island, I don't know. Maybe there is a species in there that we don't know about yet. But maybe it's just very hard to evolve toward that island, right? If you're just a sort of normal looking, not extremely curved bacterium, you have to go through the sea of suboptimality. So anyway, I guess that's all I had on there. Sorry, I've used up almost all the time, but thanks very much for listening.